Well, it is 7 o'clock. We got almost 30 folks here. A few more coming in, so I think we'll get started. All right. So good morning, everyone, and welcome to the November 2025 installment of the GCE for All webinar series. This webinar series is hosted by myself and co-host John Lewick. Unfortunately, John will not be with us today. He's out on an NIH review panel, which, again, thankfully are running again, but we will miss him today. Quick reminder for new folks, we meet every third Thursday of the month at this same time. And while the main theme here is of course, genetic code expansion, we're always excited to host speakers from different but mechanistically related fields, such as tRNA therapeutics, ribosome structure function, protein translation, directed evolution. We're always on the lookout for exceptional scientists from both academia and industry, including PIs, of course, but also grad students and postdocs who might want to make some new connections as they enter the job market. So if you know anyone who might be interested in showcasing their work with us, please do reach out to the center. Okay, today I'm delighted to introduce Dr. George Burslem, Assistant Professor of Cancer Biology at the University of Pennsylvania. Dr. Burslem is a rising leader in chemical biology and targeted protein manipulation with a research program that bridges small molecule pharmacology, protein engineering, and cell biology. Dr. Burslem completed his master's in medicinal chemistry at the University of Bristol and went on to earn his PhD in chemical biology at the University of Leeds, where he worked on new approaches for developing inhibitors of protein-protein interactions. He then trained as a postdoctoral fellow with Craig Crews at Yale, putting him right at the epicenter of targeted protein degradation research. 2020, he joined the faculty at UPenn. In just a few short years, his lab has become a highly visible hub for new technologies that expand how we edit, degrade, and control proteins in living systems. This group has made notable contributions across chemical biology, including novel molecular glue degraders, intracellular protein engineering tools, and new strategies for manipulating endogenous proteins. A perfect example, I think we're going to hear a bit more about today, is this team's recent science paper on intracellular protein editing, or Dr. Burslem, with our former undergraduate and now postdoc in the GC Center, Jenna Beyer, used bacterial genetic code expansion to incorporate non-canonical amino acids into recombinant intein donor proteins, which are then delivered into mammalian cells to edit endogenous proteins through split intein splicing. Dr. Burslem, he's authored over 50 peer-reviewed papers in high-impact journals such as Science, of course, Nature, Chemical Biology, Jackson, Nature Communications. He's an inventor on 5 patents and won many awards, including the ACS Chemical Biology Early Career Award. It is a fantastic pleasure to have you here today. Dr. Burslem, please take it away.
Great. Thank you. I'm really excited to be here. I'll just screen share. Okay, can you see presenter mode or can you see?
We see, switch, you want to switch your,
Switch
this,
screens.
yep.
Is that better?
There we go. Perfect.
Okay, great. So thank you for that very kind introduction and the invitation to be here. As you as you mentioned, I'm going to talk about our new technology on editing proteins intracellularly. So a lot of what my lab thinks about is what happens after the central dogma. We all get taught this from high school onwards, or maybe even earlier. DNA becomes RNA, becomes protein. What we like to think about is then what happens. What happens to that protein is largely it gets post-translationally modified. Lots of things are controlled by PTMs, and this massively increases the number of proteoforms in a mammalian cell from those that are genetically encoded to those that actually exist. So a lot of what we've done over the last few years is try and develop improved tools to study PTMs, both in chemical epigenetics, induced proximity, intracellular protein editing, which I'll talk about today, and ubiquitin chemical biology. We also study the biological roles of post-translational modifications, particularly in protein degradation, how metabolic states control PTMs, and then some other types of PTMs beyond lysine acylations, palmitoylation and kinase chemoproteomics. And so what I want to talk to you about today is a project that, as you mentioned, Jenna, who was trained in the GCE Center and is back in the GCE Center, worked on in my lab, which was when we think about biology, genomics informs us about DNA and RNA, but really tells us nothing about protein and protein post-translational modifications. And so one of the things we think about is how do we measure those things, and we do a lot of proteomics. But crucially, CRISPR also allows us to edit DNA. Um, but we kind of thought, how cool would it be, and what could we enable if we could edit protein sequences and protein post-translational modifications? And we kind of also wanted to take the tools that we have in chemistry and be able to apply them inside living mammalian cells. And so we set out to think about how we might be able to do this, and we wanted to have temporal control, site specificity, and crucially not necessarily been limited to the canonical genetically encodable elements. Um, so can we make unnatural proteins inside human cells? And so we came up with this idea, which was certainly high risk. I think I'm on record as calling it, frankly, insane. But we decided to try it. And the idea was that we could use genetic code expansion in E. coli to make proteins or peptide sequences that we could do chemistry on in purified form outside of cells, and then deliver them into genetically engineered mammalian cells that were engineered to contain a kind of a landing site or an acceptor sequence. So we call these the donor and the acceptor sequence. And the idea was that we would use split inteins, which are these naturally derived split proteins, which, when they're in the same space, interact with incredibly high affinity, and then autocatalytically perform a reaction where they splice the intein sequence out and join the peptides on either side of them together to form a new native peptide bond, giving you this new polypeptide backbone for a protein and a spliced product. So the idea that we had was to put these together, two orthogonal pairs of inteins. You can see them color-coded here, and also with complementary shapes. And the idea was if we had two pairs of those, we could do a kind of copy and paste reaction or cut and paste reaction and splice a sequence that we can do chemistry on from outside of cells into cells, and therefore edit the sequence of a protein and incorporate non-canonical amino acids. I vividly remember pitching this project to Jenna and saying, we can try it, but when it doesn't work, after 6 months, we have to do something sensible. And she said, okay, well, I'm game to do it. And so we did it. And thankfully, it worked, in no large part to Jenna's dedication and excellence in this type of work. So the first thing we did was try a bunch of constructs and try and figure out what the right intein pairs to do were. I'm going to skip over that and show you the first real key protein experiment. So we engineered a cell line by overexpressing calnexin, which is an ER localized protein. But the cytosolic face of it contains these two split inteins. So we have a GP41 N-terminal domain. We have a part of a split GFP and a MIC tag for tracking. Then we have the C-terminal domain of the NPU-AVA split intein, and then the rest of the calnexin protein. We also expressed and purified the SUMO GP41C, HA, AVA-N histag protein, which we could purify in E. coli, and we deliver that into cells by electroporation. And we were really pleased that we could actually then see exactly what we had hoped to see, a high molecular weight HA immunoreactive band, exactly where we would predict it. We saw loss of the mix signal as we've edited the sequence to remove the mix signal, and we see a change in the calnexin because we've reduced the size of it, but we've retained the C-terminal tail, and we just basically have swapped all of that machinery for a simple HA immunoreactive tag. For a long time, I'm sure I drove Jenna insane because I was convinced we were kidding ourselves and that this was too good to be true. Eventually, she convinced me by doing the editing, doing a pull down and purification, and then running mass spec across the splice junction, and we were able to kind of prove that exactly the peptide sequence that we had hypothesized was being formed. We got a really nice spectra of this peptide across both splice junctions, and exactly what we were hoping for was happening. And then we could finally kind of breathe a sigh of relief and actually believe that this was real. And then We decided to see how fast this was. As I said, we wanted it to be rapid in vitro. Intein pairs go in splice in on the timescale of seconds to minutes. We thought it would be slightly slower in mammalian cells, but we were wrong again. So actually, basically, as fast as we can run the experiment, the reaction seems to be complete. This is 10 minutes after electroporation of the donor into those same cells. You can see that new immunoreactive HA band at about 100 kilodaltons. You see the donor being delivered and then being degraded because it's an intrinsically unstable protein, and you can see almost complete loss of MYC and a complete drop in the molecular weight of calnexin. And so we think that we're getting very efficient editing of calnexin in mammalian cells in very, very short order. Of course, a technology like this is not useful if you can only use it to study calnexin. So we decided to probe how generalizable this technique is. We first turned to actin. We made two different acceptor sites in the actin protein, actin B, and we're able to show editing, again, using the same donor here. This is a generic donor that just inputs a HA sequence. So we can track it. Only when we deliver the protein and only when we electroporate can we see this editing occur. It's been quite effective at the intein-2 site. At the 5 site, it was incredibly efficient. You can see kind of almost complete editing there. We then wanted to try a kind of a more bespoke sequence, and so we decided to try a kinase. So we made cell lines that expressed the kinase acceptor sequence, and we expressed and purified a specific donor for CHEK1, a DNA damage response kinase, that we can then edit that together and basically result in the native sequence of CHEK1 being assembled in cells. We're able to do that. We can see that we get edited CHEK1. It's very robust, and we can actually, in the paper, we showed that this protein is not only being assembled, it's also functional in cells. So we can do kinases, we can do structural proteins, we can do ER localized proteins. We thought we should probably do something in the nucleus too, just to get the full set. So we did a transcription factor, and we did c-MYC. Here again, this is a bespoke donor which contains the Degron sequence of c-MYC. So once it's assembled, it again looks very similar to the native c-MYC, but then gets degraded. We wanted to use this as a tool to perhaps study protein degradation. Again, it works in the nucleus, it works for transcription factors, and we can see this working quite nicely. Again, just to prove that it's non-perturbing, we did interactomics mass spec on the MYC, and we're able to show it still forms the same protein-protein contacts. It still heterodimerizes with MAX, and it still seems to share the most of the interactome you would expect. It has the same half-life as endogenous MYC. As you can see here, it's about 40 minutes. Crucially, this enables us to do pulse chase experiments to study protein turnover without perturbing the system with something like cyclohexamide to stop new translation. So we can both see the degradation of MYC and the re-expression of MYC from the genetically encoded sequence at a different molecular weight. So the acceptor is depleted when we deliver the donor. We can then track the degradation of the complete c-MYC, but at the same time, we can see re-expression of the acceptor sequence, proving that we can do some kind of cool pulse chase type experiments with this technology, and really that we have this exquisite kinetic control. And we also did cut and run with this to make sure that the edited MYC ends up at the same sites at the genome, is still able to act as a transcription factor, and indeed it does, both with endogenous MYC and with overexpressed flag MYC. You end up with the peaks all in the same places, exactly as you would have hoped. So that kind of proved to us that we could do this in a minimally perturbing way. We could do it on multiple proteins, but of course it doesn't actually use the power of genetic code expansion yet. So what we then did was express and purify our donor in E. coli with resources from the GCE for All Center. to incorporate this phenylalanine. We were able to express large amounts of this after extensive optimization of the expression conditions. And then we can do all sorts of chemistry on this donor sequence. So we can do click chemistry, either copper-mediated or strain-promoted click chemistry, and incorporate fluorophores of different colors, biotins, post-translational modifications like GluckNat. We can also put triglycine sequences for ubiquitination from another technology that was developed in the lab, and super resolution compatible fluorophores. So we can make all of these things from one part of protein and do it all biochemically, purify these things so there's no excess dyes being added to the cells to give you issues with background in imaging, for example. And then we can deliver these into cells in exactly the same way, and by altering the cargo on the intein donor, we can basically determine what we incorporate into our protein of interest. Here you can see we can do the HA tag that I already showed you, but we can also do TAMRA or a biotin tagged protein. It doesn't seem to affect the activity or the efficacy of the editing. And then we can do things like imaging in TAMRA that's localized to the protein directly. We know exactly where on the protein it is. We know exactly, the stoichiometry and the protein. There's one fluorophore per protein. There's no antibodies required here. And so we kind of started calling this immunofluorescent imaging without the amino. It's just direct fluorescent imaging of a protein inside cells. And what you can see here is that when we deliver the protein into cells that are not expressing the donor, it's very quickly cleared. There's a little visible at 2 hours, but mostly it's cleared. Whereas if we deliver this donor into actin acceptor cells, you can see it out for 24 hours and in fact longer, and we're able to do imaging of these proteins. And then we thought, could we do this to endogenous proteins? And rather than doing this to proteins that we are expressing on top of the endogenous proteins, we can actually then use an intron tagging approach developed in the lab by collaborators in the Chalem lab. And they developed a system where we can insert this cassette into proteins at the endogenous level, and that's what we did here. And so this is now editing either an HA, a TAMRA, or a biotin into the endogenous levels of calnexin. Again, you can do imaging, and you can see that the TAMRA co-localizes with concanavalin A at the ER exactly as you would hope. You only see it when you deliver by electroporation. You only see it when you have the acceptor cells, et cetera, et cetera. We can now do imaging of endogenous proteins by confocal microscopy without the use of any antibodies, or actually without the use of any fixatives or permeabilization of these cells. We can also do pull downs using the biotin of the endogenously labeled protein. And so here you can see that we can pull down biotin-related calnexin. We were able to map the interactome of this endogenous calnexin. And again, we kind of call this IP without the I. So it is really exciting. What we hope this tool will be used by lots of people for cell biology, especially for proteins where there aren't good antibodies available to do these things, but that maybe are still important to study. So a summary of the technology, I think hopefully I've convinced you that we developed a technology to edit the primary sequences of proteins inside mammalian cells by using genetic code expansion and chemistry outside of cells, and then some tricks to deliver that into endogenous proteins, making a new but continuous polypeptide chain in endogenous proteins. It enables us to incorporate non-canonical functionalities. It gives us some temporal control, within 10 minutes, and then we can see what happens. It's multiplexable in a variety of ways, in that we can use the same donor for multiple cell lines. We can use the same GCE donor for multiple labels. and we could use the same cell line to incorporate multiple different modifications to the peptide backbone. And we hope that it's going to enable antibody-free approaches to cell biology assays. I also want to mention that concurrent with our publication in Science, the Mio Lab also published a very complementary system where they used inteins to do protein editing using a coordinated transposition reaction, exactly the same technology that we used, but mostly focused on biochemical approaches rather than in cell approaches. But they're really both complementary papers. And so we think this now is a new tool with hopefully diverse applications. We're interested in using it for the temporal control of PTMs, which I'll show you something in a second. We can do chemical labeling of a variety of types inside cells on endogenous proteins. We can do in situ fluorophore labeling. We started looking at in situ cross-linking to identify what proteins are actually interacting in the endogenous levels inside cells in a minimally perturbing fashion. And we also have some ideas for switching subcellular localization, for example, editing an NES into an NLS by editing the primary peptide backbone. Now, one of the major criticisms that we've received from this was that people hate electroporation. We love electroporation. We think it works really well. And once you've optimized it for your cells, we don't see a lot of toxicity, but we appreciate that that's not true of all cell types. So another thing that we did was develop a protein version of this that's deliverable by lipid nanoparticles and formulate these into LNPs. You can see here it took some modification of the peptide of the protein donor. So we use the D18 version of the donor, but actually once we do that, formulate them into LNPs, we get very efficient editing of proteins. And in this case, there's no electroporation. We just add these LNPs to the media and observe editing. You do lose some of the temporal control, but you certainly gain in the ease of use and the lack of electroporation if your cell lines can't tolerate it. And then, as I mentioned, a little sneak peek of some of the stuff we're working on now is editing PTMs in mammalian cells. And so again, we've been very much enabled by the GCE for All Center and been using this permafos set of reagents from the Mel Cooley labs to develop intein donors that contain non-hydrolyzable phosphorylation at key sites on kinases. We were able to take our intein donor. We can prove by gel shift, by Western blot, by mass spec that we have non-hydrolyzable phosphoserine site-specifically incorporated. And then we can do this and deliver this into mammalian cells and site-specifically incorporate phosphorylation onto a protein inside living mammalian cells. And what you can see here is editing of phosphorylation at serine 317 into CHEK1 in living mammalian cells. And so we're really excited about the things that this potentially enables and how we could use this to study a variety of kinase signaling events and other post-translational modifications in human biology. And so with that, I will wrap up and acknowledge the people who did the work rather than me sitting in my office talking about it. And so obviously Jenna led this effort. She's a fantastic graduate student in the lab. She was supported by a whole team of people, Nicole, Altaf, Ophir, Jay, in the Shalem lab, the GCE for All Center, and Caitlin is now carrying on this project and doing the phosphoserine work. And of course, we wouldn't be able to do any of it unless there were people willing to give us some money to pay for it. So I thank those people as well, and would love to take any questions.
All right, fantastic. Thanks, George. That was really, really wonderful. I have to say, I've heard the story a few times, and every time it feels like there's always something I catch that's new and different. That's makes it even more fun. My quick question for you is you talk a bit about the temporal control. Is there any value in having a system? So I was shocked at one of the first things that was sort of shocking is how quick the intein chemistry works.
Yeah.
Is there Have you thought about it, or is there any value in developing a system that's actually a little slower, so you sort of get like a slow time release of something where you don't necessarily maybe want it all turned over right away, but you just sort of like slowly deliver some active protein that sort of works over an extended period of time. Is that something worthwhile or that you thought about?
I think that's probably similar, kind of what happens with the LNP delivery,
Okay.
is that
Okay.
that will ramp up a lot slower. And I didn't show the kinetics for that, but we have done it, and it's still complete by 4 hours, but it's not complete at 10 minutes. And so I
Hmm.
think under systems where you might not want to entirely shock the system by turning it 0 to 100
Hmm.
at instantly, then that might be a way to do it.
Cool.
There are slower intein pairs that have been used, but I think that decreases the eff efficiency of the overall splicing because
Hmm.
there's time for it to kind of drift apart and it doesn't work as
Right.
well. So,
Right.
um, but yeah, definitely there's ways to tune the kinetics.
Yeah. Yeah. Cool.
George, I had a
Uh,
quick question here. What I wasn't, maybe I missed this. The ratio of donor to acceptor. I can see how if you had an excess of donor that might alter the biology because you're now adding some piece and how long does it sit around? Is it tenfold excess? Is it 1 to 1? And in theory, you could tune the kinetics by dropping the donor concentration down. And have you explored that ratio?
Yes, we have explored that ratio. We've done kind of all sorts of dose responses, and you certainly can get suboptimal editing by using a lower ratio of donor to acceptor. We didn't worry too much about that in the initial experiments because the unreacted donor that's delivered gets cleared very quickly. The inteins are intrinsically disordered proteins and seem to not want to hang around in mammalian cells for very long. So if you
Okay.
electroporate them in within an hour, they're kind of gone. Um, Now, for really short time poise experiments, that might be too long still. It will perturb biology perhaps to have that protein hanging around for an hour. Um, One other thing that I should have mentioned that we can do is tune PTMs by pre-mixing the ratio of PTM modified and unmodified proteins. And so we can deliver
Right.
50-50 modified versus unmodified and then get about 50% edited with a PTM versus without a PTM. So that's another way to kind of control dosing.
Beautiful.
Yeah.
Thanks.
Cool. We got a few questions from the audience. We got three. Maybe we have time to answer one or two of these. First one from Chindan Sony asking, how are these intine donor acceptor pair? How do they recognize each other? And is it possible to modify the interface between them and generate multiple orthogonal protein engineering machineries for more complicated protein engineering?
Yes, it should be. There are multiple other orthogonal pairs that we have been exploring. Um, we use the pairs I talked about today, the GP41, the AVA NPU pair, for most of this study. In some work that currently is only in Jenna's thesis, but will hopefully see the light of day before too long. We have some other versions that also work, and that was kind of by altering the the degree, the which inteins and where they're split and where they work.